I will tell you something about glycans and how we can use them as modifiable biomarkers, but also in the same time functional effectors of age-related diseases. And I will start with something we all know, but many people forget. And this is that there is no standard human. And when Alan Roses publicly said that the majority of drugs do not work in the majority of people, at that time he was the president of genetics at GlaxoSmithKline, this sent shockwaves to the medical community. But we learned to accept it, and now we know that the majority of drugs do not work in the majority of people. But to know in which people which drug works, we need biomarkers if we want to avoid trial and error. But something what majority of people are still not aware is that the same is true for the lifestyle medicine. The majority of lifestyle interventions will not actually work in the majority of people. We have to find the optimal lifestyle intervention for a given individual, for a given patient that you are talking at a specific moment. And for that, we need biomarkers. And the best class of biomarkers in this respect are glycans, despite the fact that they are the most neglected molecules of cellular communication at the moment. Glycans are the ultimate layer of molecular complexity. Glycans, as you can see on the right-hand side of the panel, can completely change the function of a protein. If you touch one glycan structure to IgG, it will be pro-inflammatory. If you attach another structure, glycan to IgG, it will become anti-inflammatory. And this is something you can change post-translationally. You can even change it after the protein has been released into circulation. Majority of proteins are glycosylated. The one revolving in the middle is the S-glycoprotein of SARS-CoV-2 virus. So everything hairy on the surface is a glycan. Trying to understand function of any protein without understanding glycans will fail. So the best analogy I can make are two birds on the left, one complete bird with the feathers, and the other one without a single feather. So protein, which is only polypeptide part, is very similar to a bird without a single feather. And you can be the best scientist in the world, and you can have the best designed experiment and execute it perfectly, but if you are using a featherless bird as a model, you will never be able to understand how birds function. So the closest to the function of a bird you would be able to come is something like this: featherless bird walking. Flight would be beyond your comprehension if you study proteins without glycans or if you study bird without feathers. But one word of caution, because people often confuse glycation and glycosylation. Glycation is a random damage. It's a chemical reaction between glucose and proteins. We quantify it as HbA1c or advanced glycation end products, but this is something completely different. This is the random damage. Glycosylation is sophisticated modification, which adds additional layer of complexity between genes and proteins. Bacteria have 5,000 genes. We have 20,000 genes. We are not only 4 times more complex than the bacteria. And sometimes when life became multicellular, glycosylation started to play the major role in modifying proteins and giving them all possible functions they now have. So polypeptide part, encoded in our gene, it can be changed only by a random mutation, and then through selection, in the next generation, it can make molecules different. Glycosylation, glycan components, encoded in a network of dozens and hundreds of genes, inherited as a complex trait, and can make proteins different either by reshuffling the allelic variants, or through epigenetics, or even through direct environmental factors. And this is something we demonstrated quite clearly when we looked at the collaborative cross-cohort of mice. So starting from the 8 founder strains, hundreds of strains were developed, they were inbred, and when we looked into these strains, some of them clearly separated from the rest based on their immunoglobulin structure and function without a single mutation being involved. Only reshuffling of allelic variants which made these mouse strains different. And we have the same thing happening with humans. Different people have very different protein glycosylation, which makes them structurally and functionally different, which you cannot see if you do not look at glycans. So people seem the same. Even the blood groups are chemically glycans. So we know that glycans make people different, but many people still ignore glycans. The reason they ignore them is that they are chemically complicated. They can have different glycan structures attached to the same glycosylation site. There can be multiple glycosylation sites. So it really takes many chemical steps to quantify it, especially in a high throughput. And my lab went there first. We did our first high throughput glycomic studies 15 years ago. So far, we analyzed over 200,000 people in collaboration with some of the best clinical epidemiological colleagues all around the world. In a recent review, which listed close to 200,000 published glycomes, revealed that we generated over 80% of them. So most of the glycomic data which is out there came from our lab. We did it in collaboration with some of the best researchers, which enabled us to push glycomics to meet the other omics, genomics, epigenomics, metabolomics, and now we are able to really publish in a reasonably good journals. And one of the first things we looked at was, of course, genetics. That's so easy, combine genes with anything else. And we realized that regulation of glycosylation is extremely complicated. There are dozens, even hundreds of genes which work together to make a decision which kind of glycan will be synthesized and attached to a specific protein. And these genes which regulate protein glycosylation are known risk factors for dozens of diseases and conditions, including the ones I would expect, like the lupus or type 1 diabetes, where we know antibodies are important, but also for the cholesterol level, for Parkinson, for some forms of dementia. So glycan seems to be involved in many different processes. And one of the most interesting ones is aging. So when we started to look at a large cohort, this first was a problem for us, because if the patients and controls were not perfectly age matched, which is not easy, there were always significant differences. And then we started to use this difference and actually track aging by looking at glycans. So what you see here is a relatively recent publication on over 10,000 people, where we, in the different parts of the world, looked how glycans in people change with age, and you can see it changes significantly. And it also enabled us to develop a commercially available glycan clock of biological aging, for which, of course, I am heavily conflicted. I am inventor, I'm one of the shareholders of Glycan Age. So I'll do my very best to only present the data and you make the conclusions. Of course, being involved in a company also enabled me to persuade the company to sponsor this meeting. So thank you, Glycan Age, for sponsoring. And also you can get a discount if you go to their booth. So good thing about the Glycan Age is that we are measuring molecules which regulate low-grade chronic inflammation. And we know how they change with age. So by looking at the molecules at the given moment, we can estimate how much you have already moved from a young and mostly non-inflamed condition to the highly low-grade systemic inflammation, which is usually linked with the majority of diseases. And this is now the Glycan Age clock, which is commercially used, actually the only clock which is being used commercially to monitor lifestyle interventions. And of course, the question is, do we need that another glycan clock? There are epigenetic clocks. There are so many different clocks. And I will not go into detail here because I don't have time. I will only make one comment, and this is that as long as we are getting 2 or 3 new epigenetic aging clocks every year, this means we still don't have the best one. We still have to wait for epigenetics to deliver on its promise. And if you're interested, in 2 weeks in Croatia, in the beautiful city of Rovinj, we'll have a longevity symposium. Steve Horvath and I are organizing it to celebrate the 10 years of genetic and glycan clocks, and you're all welcome to join if you're interested. So how can we develop a molecular clock, a navigator, a tool, which will help us to navigate lifestyle medicine and to make personalized decisions which will work for each of us? And it's relatively simple. We start with large studies, to identify links. Then you have to do the independent studies to replicate whatever we see. We have to go into longitudinal studies to see for individuals how does it change, intervention studies, and finally randomized clinical trials. And I will show you some of the things we did in this respect. But before I move in that direction, I will just emphasize again that these glycans which are attached to IgG, they're not just biomarkers. They're not just molecules which are telling us something. They're functional effectors which regulate inflammation. If you attach one type of a glycan, the ones on the bottom on this slide, you'll make IgG pro-inflammatory. It will promote inflammation. But if you attach glycans which are on the top of this slide, they will become anti-inflammatory. So the same molecule, immunoglobulin G, will either promote or suppress inflammation depending on the type of glycan which is attached to it. And this is only one of the aspects how immunoglobulins function, because they also interact with the multiple other interactions between the cells and the immune system. Excellent review was published a couple of years ago, so if you're interested, have a look. Another important aspect of glycans is that they integrate genetic information, epigenetic information, and the environment. So genetic information is our evolutionary wisdom, millions of years evolution written in our genetic code. We got it from our parents, we'll give it to our kids, something we do not change. And approximately 40% of the glycan age is defined by this genetic component. Then we have the epigenetic component, which more or less goes to our grandmother and then our mother and everything which they experience and then encoded in our epigenome while we are still within our mothers, and then everything we did in our life also is changing the epigenome, and it's also part of the glycome which we see today. And then we have the metabolic element, what we eat, how we feel, how we sleep, which all integrates together to generate glycan structures. And a kind of a big picture is, in medicine of today, usually you see a patient when something is fallen apart. There is a pain somewhere, patient needs help, asks for help, and most of the time this is beyond the point of no return. The patient gets a diagnosis, some of the chronic diseases, and becomes the regular customer of the pharma industry for the next 20, 30 years. But the disease is not a 0-1 process. It's a long molecular process of changes when people from the healthy condition change into a disease. And if we can identify molecules, which we can quantify very early, then maybe by a simple lifestyle intervention, we can prevent this endpoint which we would like to avoid. And glycans are excellent here because they change up to a decade before that disease becomes diagnosed. We did dozens of diseases, and in the majority of diseases, especially those where we know that the chronic inflammation is one of the components, we see glycans changing in the same direction. They change in aging, but accelerated. So accelerated glycan aging is something which happens up to a decade before a disease, and then depending on the genetics, epigenetics, and whatever, it will end up in one of the diseases which are linked to inflammation. So I have no time to go through many examples, but I will show you an example of cardiovascular diseases and what we have done so far in using glycans to predict risk and potentially even decrease risk of cardiovascular diseases. So initial study was done 6 or 7 years ago. We did it together with Tim Spector and Jim Wilson, over 3,000 people, cross-sectionally looked at the glycans, looked at the cardiovascular disease risk score, and saw a correlation. It replicated perfectly, so we said glycans associate with the cardiovascular disease risk score. Then we looked into the longitudinal cohorts, and this is something we did with the Tim Spector, 2,000 twins in 3 time points over 15 years. So we could see when the hypertension developed and when the glycans changed. And the change in glycan came before the hypertension was developed. We went in the large prospective cohorts, collected 30 years ago, samples collected, clinical follow-up. So we had a sample, and then we knew who will have a heart attack or stroke in the next decade. And the glycans in men were as predictive as the entire AHA score. In women, even single glycan was more predictive than the AHA score. Interestingly, completely different glycans predict heart attack and stroke in men and women. So there is a difference in pathophysiology in respect of glycans in developing cardiovascular diseases in men and women. This was a large study, a couple thousand people, published a couple of years ago. But one study doesn't mean anything. We did a replication study. Again, a couple thousand people collected a long time ago. Clinical follow-up. Again, glycans predict heart attack and stroke, different structures in men and women. And the hazard ratios were actually not that small. So there is a significant risk prediction in glycans, sex-specific. And then we went for another 2 replication cohorts, this together with the colleagues at Harvard. This was still not published. This is just the abstract and a presentation which was published in Circulation. And then we went into animal studies to see, is there a molecular mechanism we can explain? Is this something we can actually modify? And this paper published in Circulation a few years ago clearly shows that if you have a mice, put them on high-fat diet, they become obese, they will develop hypertension. But if you feed them with a dietary supplement, which is improving IgG glycosylation, they will, of course, become obese, but they will not develop hypertension if you protect the IgG glycan, which is suppressing inflammation. So there are molecular mechanisms and multiple replications showing that the glycans we measure today can predict and possibly even contribute to the disease development. So the key question here is, can we revert this glycan clock of aging? Can we make an actual decision to do something, to slow it down, or even completely stop inflammation? And from our genetic association studies, we know that there are at least 16 genetic loci which are involved in the regulation of the glycan age clock of aging. And actually, this is a med-archive paper, but it was accepted in Aging a couple of days ago, so it should be out very soon. And we did a function validation of these heads using a special epigenetic CRISPR tools we developed. So we know which genes are functionally important. In theory, which genes could also be druggable at one point, but at the moment, let's only talk about the lifestyle intervention and see what can be done. Because, of course, drugs can change things. If you take strong anti-inflammatory biologicals like infliximab, you will change IgG glycosylation. Not in everybody, but in the majority of people, anti-inflammatory biologicals change IgG glycosylation, but this is not preventive medicine. You don't want to do that. Also, another very important aspect of IgG glycosylation are the hormonal factors, especially in women. If we look at the change in IgG glycosylation with age, and on this slide, men are blue and women are red, we can see that in the reproductive phase, females are always kind of protected from these pro-inflammatory effects of glycan aging. And then there is a rapid acceleration around the age of menopause, and then they catch up with men. And this is independently replicated in 5 populations from China to Europe. And on an individual level, it can be really dramatic when somebody would have a reasonably good glycan age, and then we had women aging for 20, 30 years within a year, which of course made them very angry at the moment, told us, you know, your test it's not good, it's giving horrible results, but eventually they realized where the problem is. And we did a proper placebo-controlled randomized trial showing that it's estradiol, which is causative, which is causing changes in IgG glycosylation, both in women and men. So in men, the testosterone was given, but testosterone had to be converted to estrogen to be functionally relevant. And we even believe that IgG glycan composition could be used in a very similar way as HbA1c is used today for glucose, because HbA1c is integrating glucose concentration across a couple of weeks. IgG glycan composition is integrating estrogen concentration in a couple of weeks, because the problem of perimenopause is that the hormone levels are fluctuating, and you cannot really, by measuring hormones, tell anything. But if you measure IgG glycan composition, you can say what is the average hormone concentration the last couple of weeks, and it can be very indicative of the early stages of perimenopause. This usually happens before any other symptoms are visible. Something we also know that works is a weight loss. So people undergoing bariatric surgery can have a very rapid reduction in the glycan age. We had a patient losing 37 glycan age years in 6 months. Not everybody loses that much, but majority of people after bariatric surgery really do see improve in a glycan age. But again, bariatric surgery is really a drastic solution. You don't want to do it as a routine approach. So we looked what happens with the normal weight loss and weight gain. Again, with the Tim and 2000 twins across 15 years, what we saw is that twins who were gaining weight were aging faster than twins who were losing weight. And this was happening in the 30s, in the 40s, in the 50s. So anytime you decide to lose weight, you will actually improve your glycan aging and decrease this pro-inflammatory effects of higher glycan age. Caloric restriction more or less works in everybody. This was a study on 1,000 people who were on 800-calorie diet for 2 months. Personally, I think 800 calories a day is torture, and nobody can actually maintain this kind of caloric intake for a long time. So after the short period of caloric restrictions, they were put on 5 different diets. This was a Diogenes study done in a number of centers in Europe. And what we learned here is a very clear example that there is no standard human. So each of the diets that was studied, including the high protein, high glycemic index diet, was good for some people and not good for some other people. Also, the low glycemic index or the low protein diets were good for some people, not good for other people. So each of us is different. There is no magic diet. There is no solution you can give to everybody. You have to find a solution for every given patient. The same goes for physical activity. We had 1,000 people exercising, 1,000 middle-aged, slightly obese people exercising for months, and majority of them actually got worse because they were overtrained. You cannot just put a couch potato on a treadmill and say, keep running until you get better. You have to optimize physical activities. Only when the physical activity is optimized, you have beneficial effects. Microbiome is also an extremely interesting component of life. We know how important our microbiome is, and it's talking with the glycans all the time. Actually, the microbes are the first organisms that we domesticated because we are feeding bacteria with the mucins in our gut. And especially when there is no other food in our digestive tract, bacteria are dependent on grazing on our glycans to survive. And there is an evolutionary symbiosis between us and the bacteria we feed. And we know that, for example, if you do a microbial transplantation, you change your glycome. We know that there are correlations between the glycome and the microbiome. And we do a lot of studies trying to individualize diet based on the microbiome. We do it together with Tim Spector again. And yes, glycan age can be modified. So I'm confused with the time. It says 34 minutes, but I'm speaking, I should finish now.
4 minutes.
4 minutes, oh,
3 minutes.
4 is plenty. So we know that people who do interventions can change. This is Tim Spector's story. He managed to lose 11 glycan years with interventions he did a couple of years ago. He even recorded an Instagram Live about it, so if you're interested, you can see it. And he's also mentioning it in his latest book. We have anti-aging clinics or longevity clinics. Some of them are extremely efficient. So Glycan Age together works with over 700 clinics all around the world. Some of the clinics hate Glycan Age because Glycan Age shows that they're selling the snake oil. Some clinics love Glycan Age because the Glycan Age shows that what they're doing is actually helping people. So if you want to really help people, you want to have a tool to quantify, to show to your patient that you're actually doing something good. If you're just making money on your patients, then you don't want to have any kind of independent evaluation. And kind of summary of the whole story is, we know that we have to do preventive maintenance on our cars. Nobody will change tires only after they explode on a highway. This would be very deadly. But if we talk about our body, we rarely do preventive maintenance. We wait for something to explode before we ask for help. And this is something which has to be changed. So we have to start monitoring our health as soon as possible. We need to develop different biomarkers which will tell us how this trajectory is going, intervene early, and change. And one of the key problems we all have is that people do not do what is known to be healthy. We know what are healthy habits. People don't do them. One of the key reasons is you don't have a feedback. If my feedback will come back in 20, 30 years, I will not change my lifestyle today. Sure. But even these wearables, if they give us feedback, they have some effect. So if you objectively quantify, you have a blood biomarkers which will tell you, you're doing good, you're doing bad, this is not working for you, try something else. People are more prone to change their lifestyle. And I do believe that the biomarkers we are developing are giving objective insight into current state of low-grade chronic inflammation. They're the quantitative tool which can help evaluate lifestyle interventions and then give you fast and objective feedback.